Okay, thank you for the opportunity to speak about this work and present some very hot off the presses results from myself and my co-author Sam Bartels, who's a postdoc working with me, and Laureen Echeverry, who's a master's student with me, and Sylvia Ronzani, who's doing a PhD with John Spence. And I'm going to talk about biodiversity and WAM at a slightly different scale than Scott talked about. So I think our work is quite complementary. So why did we ever think to start a project on WAM and biodiversity? Maybe it's obvious by now, but I think the point is that moisture is a major driver of forest ecosystems and lots of other ecosystems across the landscape. So we imagine it to be a major driver of vegetation and therefore of other biodiversity. And the question that we were asking really was whether LiDAR data and WAM could be used to predict biodiversity. And then we're also looking at forest productivity and carbon dynamics. And then secondly, a response of these to variable retention harvesting. So we're looking at, I'll be presenting results on vegetation and beetles today. So I think Scott was looking, if you take a landscape like this and go across the entire landscape, and Scott was going across the entire landscape looking at the whole diversity of ecosystem types or forest ecosystem types that we could. For this work, we were at a slightly finer scale and a narrower scope, so we were confining ourselves really to these upland sites that are varying in composition, forest composition, as you'll see, but not going as far along the gradient into the really wet sites or fens or peatlands or anything like that. Okay, so we're working at the EMEND experiment. If anybody in the room is not familiar with EMEND, I'll just give a really brief overview for it. So it's a long-term adaptive forest management experiment that's been going on for nearly 20 years up here northwest of Peace River. And the experiment encompasses 4 different forest cover types, so a gradient of mixed wood composition from broadleaf or aspen dominated, which I'll now have to call deciduous dominated because I like broadleaf, but everybody else calls it DDOM. Deciduous dominated forest, and then a deciduous dominated forest with conifer in the understory, then a mixture of the two in the canopy, and then a conifer dominated forest. And across these different forest types, we applied variable retention harvesting, 6 different levels of variable retention harvesting and unharvested controls, and have been collecting core data in 100 different compartments and 600 different permanent sample plots from the pre-harvest time period, which was 1998, up until 15 years post-harvest, which is really a little bit more than 15 years post-harvest. We did the vascular plants last year and hoping to do the bryophytes this year. So there's a huge data set here, and we decided that we would try and leverage this data set to ask these questions about WAM's ability to predict biodiversity. Okay, so what we're using is the existing EMEND data and newly collected data from last summer, as well as some new sampling. So we wanted to cover in the WAM gradient and make sure we had good coverage across it for these areas. I'll be presenting on 3 of the 4 forest types, so the gradient from broadleaf to mixed to conifer-dominated forest. We sampled mostly the unharvested forests in 2014, and then in 2015, a few more unharvested forests, and then some of the variable retention harvesting, and that work's just in progress. And we sampled vascular plants, bryophytes, which includes mosses and liverworts, and ground beetles. So the existing EMEND plots, the core plots were mostly towards the drier end of the gradient, and that's because they were randomly located, and more of the landscape is drier than it is wetter. So when you randomly locate plots, you end up sampling what's most common on the landscape, which Scott already explained about the ABMI data. So we established new plots towards the wetter end of the gradient to fill in towards that end. And so very crudely what we did is we took the wet areas mapping depth to water and flow accumulation values and looked at them in a map like this and said, okay, in a given location, we're going to put some additional sample points representing that gradient. So we had little groups of plots like this in different compartments of different forest types that would fill in across this gradient, realizing that most of the core EMEND plots were up towards this end of the gradient. So we set these out in a GIS ahead of time, chose them, and then located them in the field using a precise GPS system, and then set them as the sample points and sampled all 3 different groups for biodiversity at those locations. Okay, so for vascular plants and bryophytes, we used 2x2 meter plots, which is the standard core procedure for data collection at EMEND, and did cover estimates to species. And then in a 5x5 plot that was around that, we did presence absence of any additional species. And Sylvia, for her ground beetles, had 2 pitfall traps per sample point, so at those same locations. So she had two pitfall traps, and then she pulled those for the data collection afterwards. Okay, so what did we find out? So these are preliminary results, super hot off the presses, like hours ago. But for vascular plants, what we found so far is that both richness and diversity were related to depth to water. And we tried the different flow accumulation catchment areas, much like you heard earlier from Ivan, that we tested which was the strongest, and it seems so far that for richness and diversity, 12 to 16 hectare catchments had the strongest relationships. The best model was actually with a log of depth to water as a continuous variable as opposed to categories, and there was a significant depth to water by forest type interaction. And so this is what the graphs look like. So we have conifer forest there, mixed wood forest there, and deciduous forest there, and you can see really not much relationship or possibly a negative relationship. This is for vascular plant species richness, and then this strong positive relationship with the conifer. So remembering again that these negative numbers here are the smaller depth to water numbers is wetter. So lower vascular plant species richness towards the wetter end of the gradient and higher towards the drier end of the gradient in the conifer forests. And then in the other 2 forest types, not much happening in terms of relationship. So conifer richness lower on the wetter sites. In terms of the Shannon Diversity Index, the same kind of relationship here. Little hint here maybe that we're getting the same kind of thing for diversity in the mixed wood forest, but I'm not sure that that's significant. We do have potentially some more data to add into that to see if it shows up a relationship. But for the conifer, the same kind of thing we saw with richness we're seeing with diversity. Okay, for bryophytes, again, both cover and richness were related to depth to water. And in this case, the strongest relationships was with the very small catchment flow accumulation area. So this is kind of interesting that different species groups seem to be more strongly related depending on which catchment you use. And for bryophytes, it was either 0.5 or 1 hectare catchment area. Again, significant effects of depth to water and forest type, and a significant interaction of depth to water and forest type. Looking like this. So what you'll see immediately is that this is for bryophyte cover. And again, this is the wetter end of the gradient, the lower depth to water values. And you'll see for cover the opposite relationship that we saw for the vascular plant richness and diversity. So here we have increasing cover of the bryophytes towards the wetter end of the gradient. Okay, deciduous dominated forest, not much happening. Mixed wood forest, maybe a decline in cover towards the wetter end of the gradient. I'm not sure that's significant, and there's a lot of outliers here. You'll notice that there's not much in this area here, and you'll also notice here and here that we're lacking points towards this drier end of the gradient. That's because we're going to get that drier end of the gradient from the core EMEND plots, and those were sampled last summer, and we don't have the IDs back yet, so it takes a long time to ID bryophytes. So we're awaiting on those data there to really confirm these relationships. For bryophyte richness, interesting here, really for the conifer forest, not much happening so far with the richness in terms of depth to water, but again, we're missing all this here. Deciduous dominated, really no relationship. Mixed wood, very interesting, with looks like a fairly strong relationship of increasing richness of bryophytes, again, towards the wetter end of the gradient. And it's not super surprising. We know that bryophytes are very sensitive to climate and to moisture. They require moist sites, so expected to see this, but we sampled all the different bryophytes that were in the plot, so some were on dead wood, some were on the ground, so it's not just forest floor bryophytes that are being influenced, it's the entire community, and we'll start breaking them down into different species group and looking at composition. Okay, for the beetles, Sylvia has data for the conifer and deciduous forest. Her response variable standardized abundance per species. So what the analysis she's completed so far is an analysis of composition. So she's looking at her entire set of data, the species that she had and the relative abundances of them as related depth to water and forest types. She found that the forest types differed from one another and there was a significant effects of depth to water. And she got the strongest relationships with 8, 12, or 16 hectare catchment areas. Okay, so she has just one ordination diagram I'll show. So this is the results of a multivariate analysis and ordination redundancy analysis. So it's relating the composition of the beetle community to the forest type and to the depth to water at different catchment areas. So you can see here we have it's a little bit hard to see, but this is like the bubble that represents the conifer-dominated forest, and this is the bubble that represents the deciduous-dominated forest. And so they were different from one another, which is not super surprising. And then also these vectors here represent the different WAM gradients. So you can see this is going to be a higher value of the depth to water, so a drier end here. So again, it shows it shows the fact that the deciduous-dominated forest tended to be slightly drier than the conifer-dominated forests. So conifer and deciduous, and then some evidence of a relationship of the community composition with depth to water. And we haven't had an opportunity, or Sylvia hasn't had an opportunity to look at it into more depth yet. Okay, looking at deciduous and conifer separately now to separate this out, you can see again there is a gradient here with this being the drier end of the gradient and this the wetter end of the gradient, and a nice spread of sites across this. So some suggestion that the composition of the beetles is related to depth to water here. And then this is the same for the conifer, although it looks like depending on the catchment area that was used, that we're going in slightly different directions here, but again, a spread of the composition relating to the depth to water. Okay, so conclusions for vascular plants in conifer-dominated forest. It seemed like richness and diversity tended to be lower on the wetter sites, and maybe for mixed wood. Jury's still out on that. For bryophytes in the conifer forest, the cover was higher on the wetter sites, and mixed forest may be opposite. And interestingly, in the mixed forest, the richness was higher on the wetter sites. And we are awaiting more data. For the beetles, so far we can just say that it seems that composition varied with depth to water, and that there was differences between the forest types in terms of how this relationship played out. Okay, so what's coming up after this? Other parts of the project that I didn't have time to talk about. We will be looking at forest productivity, not in the level of depth that you heard already, but taking advantage of the permanent sample plots over time, in which we have information on the growth of the trees and the regeneration of the trees after the partial harvesting. So we'll see whether depth to water or flow accumulation can provide more information on those responses and on the mortality of the trees. Postdoc Ding Liang is here. He's working on that. There's another master's student, Paul Sewell, here, who's here, and he's looking at soil carbon. So he'll be looking at whether in these same sample plots or subset of the same sample plots that we were using for the biodiversity, he's looking at soil carbon dynamics. And then we will be looking at the responses to the partially harvested forest. So we'll see whether a depth of water can make some prediction as to how resilient or how the ability of these different forests is to recover following variable retention harvesting. Just before I say thanks to funding, this was funded largely by an NSERC strategic program grant and U of A, Alberta Conservation Association, Alberta Ingenuity Fund. I'd like to thank field assistants and collaborators, Barry, Vivi, and Nicholas, and a number of people that helped with the plant IDs, and then of course the EMEND partners who provided all the infrastructure and the pre-existing data at the site. Just before I wind up, I'll just make one little comment, because we have had a couple of comments about LIDAR and WAM in the white zone of the province. I have a little side project. where we're looking at using WAM and depth to water to see if we can make predictions about locations of invasive plants in rangeland or grassland communities. And so we have we obtained LIDAR, and the province provided us with the wet areas mapping for the Matthias Ranch. This is a 12,000-acre ranch down by Dutchess, and my technician, Lori Schrader, is here, and she's working on that project, and we're cooperating with Alberta Parks and Joyce Gould, and we'll do some work in Drumheller as well. And so it looks like that's turning out preliminarily some really interesting results as well in terms of our ability to use depth of water to predict where invasive plant species will be.